Now we will discuss a little more about detection methods of yeast and mold in different food. And we earlier discussed that some of the media are specific for certain yeast and mold as well as to certain food. For example, we talk about this DG18 or use of sucrose contents in the maltose syrup or as well as in other simple sucrose solutions as well. These agars are normally suitable for food that have got higher water activity, generally above than 0.95. That is the kind of water activity that can be seen in fruits and vegetables, meat and dairy products. So these media can be used for enumeration or detection of yeast and mold in these kind of foods. So there are some other media, particular media that are generally used in the lab. These include the dichloran, rose bengal, chloramphenicol agar. This is called as GRBC, or there's another one called plate count agar with chloramphenicol, PCA, potato dextrose agar with chloramphenicol. This chloramphenicol is basically is an antibiotic, so PDAC is used. Then potato dextrose salt agar with chloramphenicol is used for different spread molds. Similarly, there may be some other media which are used, such as peptone water. 0.1% peptone water is used. 2% sodium citrate is also used, which is tempered at 45 degrees C for food such as cheese. Similarly, we use 0.1 normal HCl and sodium hydroxide as well, and gram stain solutions are also used. So these are the kind of material that may be needed for detection of yeast and mold in different foods. If we talk about the equipment that are needed or instrument that are needed, it includes pH meter or pH paper. Similarly, we need to have light microscope to look at the morphology of yeast and mold. We need to have colony counter as well to calculate the colonies. That's an optional part because they can be visually counted as well. Then incubator is needed, and it has to be a darkened incubator because the incubation takes place in the dark here. And the temperature of incubation, as we earlier said, they have to be 22 to 25 degrees C. And we also need to use water bath for this kind of analysis in our lab. So these, as I said, the plates are incubated in dark, and then we calculate the colonies on third day or fifth day. They may be calculated on third day if we see that there are more number of colonies, and if the higher numbers of colonies, then the colonies can be counted on day 3. But in case if they don't not show up on day 3, then these colonies can be counted on day 5th. But if we look at the number of colonies on day 3, we should take extra care that we do not mess up things and we do not change the colonies or depress these colonies in a way that it can affect the number of colonies on day 5th, or there should not be any chances of any contamination or dislodgement of these colonies by touching these colonies on the plate. Now, once we have looked at all the colonies, their morphology, we have seen whether they are yeast or mold and what kind of mold or yeast they are, then we need to look at, we have to calculate this average count that has to be done arithmetically, that has to be taken a mean because we deal with more than one number of plates. And we normally use this formula, N, that is equal to A multiplied by D, and here N stands for number of colonies per gram or per milliliter of the product, and A is the average count per plate. So we take the average count per plate. If we have 3 plates, we take the average of these 3, and D is the respective dilution factor. Because we dilute these colonies or the material that has to be taken from food or surfaces, we use dilution, so dilution factor must be taken into account, and using this formula we can have a number of colonies of yeast and mold within a food product or a food surface.